Hello again, I'm Dr. Jordan Taylor, Undergraduate Exercise Science Program Director and Associate Teaching Professor at the University of Kansas. On today's episode of Fitness Facts, I have invited Dr. Andy Fry back for a detailed discussion about skeletal muscle fiber types. Dr. Fry is a professor in the Department of Health, Sport, and Exercise Sciences at the University of Kansas. He's also the director of the Jayhawk Athletic Performance Lab. Throughout his career, Dr. Fry's research has consistently focused on skeletal muscle physiology, resistance exercise, endocrine responses and adaptations to exercise, and overtraining. Welcome back to the show, Dr. Fry.
Thank you. I'm glad to be here.
All right, so Dr. Fry, tell us a bit about your background researching skeletal muscle fiber types and exercise training. You know, what's motivated you to do this area of research, and maybe some of the interesting findings from your study might highlight those.
Well, I'm going to go back long before my graduate work. For a number of years, I owned and operated a fitness center, and we sponsored competitive lifting teams, powerlifting, weightlifting. We sponsored and held bodybuilding competitions, and it was really geared toward the competitive lifter. And when I started my graduate work, of course, I kind of followed along with an interest in resistance exercise, the world of strength and conditioning. And my training at Penn State was with a gentleman, Dr. William Kramer, who's well known for his work in the hormonal responses and adaptations to exercise. And I realized that those hormones have to function on some target tissue. And so the interest of the tie-in between the endocrine and, in this case, the target tissue being skeletal muscle, really piqued my curiosity. And so my postdoctoral training was at Ohio University, where the focus then was on skeletal muscle physiology and the fiber types and the protein expression, the different adaptations, and how do you perform that kind of research. And that has led to a long series of a number of decades of research in related fields.
And this is something that, I mean, students are so interested in this. This was one of my favorite things to talk about and learn about when I was an undergrad exercise science student and then going through graduate school and everything else. It was just a hot topic. I mean, you hear, sports announcers in a football game or whatever say, oh, so-and-so is just not very twitchy, or they're twitchy. And, they're kind of making an indirect reference to muscle fiber types, fast twitch, slow twitch. This athlete's very twitchy. He's probably got a lot of fast twitch fiber. So Dr. Fry is going to get into talking about some of this and what all that means. Any interesting findings as far as your research in the area of muscle fiber types and resistance training that you want to highlight, or that were like really maybe seminal findings in the field that I guess your work that you're most proud of, maybe?
Well, there's really a couple of areas, because if you're getting into muscle fiber types, that means you're getting into what's going on in the cell. And that opens up a whole Pandora's box of different things. So that has allowed us not only to look at the type of muscle fiber that is in the muscle or the adaptations of the fiber types, but also other things inside the muscle, such as the signaling pathways that are related to basically all the functions of a muscle, as well as factors that influence it, such as the hormones that will look for the receptors. In this case, the receptors in the muscle, maybe they're androgen receptors, maybe they're glucocorticoid receptors. So it's opened up the door to a vast world of cellular adaptations. I've really enjoyed the work looking at some of the unique characteristics that certain athletes have that we think is one reason that perhaps they excel in their sport, whether it's world-class powerlifters or world-class weightlifters or high-level elite athletes in a number of other sports, and what are some of the unique characteristics. This has been studied for a long time. You can go back over half a century, and people have been looking at this and asking questions. We also sometimes look at the maladaptations. What happens when you do too much? And in this case, we have a long history in our lab of looking at overtraining, and what happens when you do too much. Not so much that you become injured, but your performance is impaired. And many people that have trained seriously may have gone through phases where They may not be fully overtrained, but something has gone amiss. They're not hitting peak performances. And can we explain part of this story by what's going on in the muscle fiber?
Yeah. Okay. Well, getting into some of your research here, and you're going to walk us through these slides. We've got a lot of interesting stuff to look at. Um, how is a skeletal muscle biopsy obtained? Can you discuss the procedure? And I'm going to hand you a biopsy needle here. Maybe you can
Yeah.
talk about that. Oh wait, and I gotta get my pants off of my leg up here so we can do one. No, I'm just kidding.
Well, we'd be happy to oblige.
Right. Well, we don't have any lidocaine or anything. I guess I'm just gonna have to, I don't know, do a shot of whiskey and bite a stick, and then you know, pop it in my thigh. So
Um, this is a minor surgical procedure, and we actually have an illustration here of a biopsy needle that is being removed from a thigh. This is a sample where, or an example where we have the vastus lateralis, one of the lateral thigh muscles, which is a favorite target because it's easily trained. It's a large muscle. It's easy to access. There are no major blood vessels or nerves nearby, and there's a lot of literature on this muscle.
so you can see that here on this slide too. That's, you said, the vastus lateralis right there.
Yeah,
Okay.
and so we get a tiny amount, and people say, well, how much do you take out? Well, it's on a really large biopsy, you might get 100 milligrams, and perhaps one way to describe it, if you have a sharpened pencil and you just snap off the lead tip, that might be the volume, give or take a little bit of what we get. In a good biopsy, we'll get, we like to get about 1,000 fibers, and it's representative of what's in that muscle. And in order to do that, we use this is an example of an instrument. This is called a Bergstrom needle, which is probably the most commonly used biopsy needle for skeletal muscle biopsies. If you talk to a medical doctor, there's a lot of different instruments that are used because biopsies are taken from a number of different tissues, whether it's bone or other soft tissues, and so they may look quite different. This Bergstrom needle has three parts. It has this inner portion that I'm going to set aside, and then it has these two parts. So this outer portion, and this I believe is a 14 gauge needle. No, it's not 14. It's larger than that, but it has a dull point. So this is not sharp like a syringe needle, but there is an opening here on the side, and this that gets inserted down the middle, this is also hollow and it has a sharp edge right here. And so when we place this inside the other, the opening is closed, and so we simply we numb up the skin, the surface area, and we push this about 1 inch into the muscle. And the person getting the biopsy will feel pressure, and when the needle is inside, we pull back on the inner portion, which opens the window, and it allows some tissue to go in, and then you snap down on it and it clips it off. And we'll quite often open it up again, turn it, and clip again. So that doubles the amount we get. And then we pull it out. And quite often there's a little opening at the end that you apply suction to, because that helps to pull the tissue in. And then afterwards, you need to get the tissue out. And so you take it over to the microscope and the material that you're going to mount the sample in, and it's inside this inner part of the implement. This little probe here simply pushes it out. And so then you can mount it, you can get it out, you can arrange it so that you have all the fibers in one direction. You can place it on either a piece of cork or a little piece of wood. We often use a tongue depressor and mount it so that it's oriented in a manner. We freeze it either in a liquid nitrogen mixture or we freeze it in isopentane. And what you freeze it in is really important, because if you freeze it just in liquid nitrogen, it slows down the freezing process because there's a little bit of oxidation. And if you're trying to get a nice section to look at the cross-sectional area and stuff, you're going to introduce some freeze damage. For other purposes, like just looking at protein expression, doesn't matter. But for the nice clean images, and we'll see a few of them here later, you freeze it in isopentane that's cooled down to the same temperature, and there's no oxidation process, and the snap freeze process is really fast. And so then you put it in the deep freezer until you're ready to do the analyses.
Yep. Cold, what, -80?
Yeah.
Yep. So let's, now that you understand how this tissue is obtained, we got this little chunk of muscle. What's the process? If we go to slide 3, how the tissue is processed, what are the different staining methods that have been used to evaluate skeletal muscle fiber types? So maybe walk us through some of these different stains that are utilized to visualize the skeletal muscle fibers, the individual cells.
So the images that we'll see here are taken from a book by Richard Lieber. He's a well-known skeletal muscle physiologist, and I really like the images. The histology is really good, and anyone who's studying histology, there will be a chapter in there on skeletal muscle. And what some people don't understand is there are many ways to stain and identify muscle tissue, and we'll look at several of them. I mentioned how we freeze the tissue and we try to arrange it where the fibers are all in one direction. And one of the next steps is we put it in, it's essentially a meat slicer, but it's called a cryostat.
Yep.
If you go to the deli, they can cut your meats and they do a real nice job and they set the thickness. Well, that's
like a giant cryostat that you see at Subway or some sub sandwich shop. Yeah, it looks
and
similar.
the difference here is these instruments, you can set it to so many microns, which is a millionth of a meter. And so a lot of the work we do, we'll section it 12 microns thick.
Yep.
And so if you breathe too heavy, you'll blow it away.
Right.
And it's that thin. But then we get a nice cross section of the frozen tissue. And so we see on this slide, this is they call it an H&E stain. It stands for hematoxylin and eosin. Very commonly used. All this does is tell us where the muscle cell is. It doesn't tell fiber types. It just shows an outline. And so in this image, which happens to be from rat tissue, you can see the outlines of the fibers. And in all of the images we'll see, they've gone ahead and identified several of the fiber types because the sections that we'll see, Lieber took one slice and stained it with H&E, and then the next slice he stained it differently, and the next. So we'll see the same fibers again and again.
And talking about those sections, like you said, you typically get 12 micrometer or 12 micron thin. That's thick, thin sections. And just for a frame of reference, the diameter of the average red blood cell in your body is about 5 microns or 5 micrometers. So these are 12. It's a little more than double that, but they're thin sections.
Yeah, and it's a tedious process, but you get it on the microscope slide, and now you can do the staining. If we go to the next
image,
this is another way to stain. This is actually for dystrophin, which is also not a fiber typing method, but dystrophin is an important muscle that you find around the periphery of the muscle cell. And so on this image, it stains dark, and so you can see really clearly the outlines of the fibers. We don't know which ones are fast twitch or slow twitch. We just see very clearly. And dystrophin is a protein that's, among other things, is associated with certain types of muscular dystrophy. When you get an improper, the dystrophin protein is incorrectly put together, then you have a problem and the muscle loses some of its integrity. And that's one of the characteristics of certain muscular dystrophies.
That's what you see like in Duchenne's muscular dystrophy. The dystrophin's kind of helping hold that cell together. And unfortunately, due to that genetic disorder, the muscle cell basically gets replaced with connective tissue and fat. And not a fun process, for sure. And it's very unfortunate because, I know it generally affects boys because it's an X-linked condition, but they're in wheelchairs or at a very young age. But I guess you want to go to the next one?
Well, I was just going to make one more comment too on the dystrophin stain. It says subsarcolemma. Sarco refers to muscle or flesh. And if you study cell biology or any general biology class, the plasma lemma is the cell membrane. Well, the sarcolemma is a muscle cell membrane. So it's just inside the membrane of the cell.
Yeah.
But the next image you see is a stain for an enzyme called myosin ATPase. And ATPase is really the fuel for the muscle cell for muscle contraction. And well, ATP is the fuel, and this enzyme, myosin ATPase, is the enzyme that breaks down ATP,
Allows the fuel to work.
allows the cleavage of the phosphates,
Mm-hmm.
which are high energy bonds. And this enzyme, myosin ATPase, is located right in the cross bridge. So if you've had any basic muscle physiology or exercise science classes, you know you have the myosin filaments and the actin filaments,
Mm-hmm.
and the myosin has cross bridges that will reach over and
Yep.
bind to the actin and pull on it and cause the muscle to shorten. And that's one of the functional features of muscle, among other things. It's designed to change shape to produce force.
Right.
And
We have to convert chemical energy into mechanical energy to do that. And
and it
myosin ATPase is what catalyzes that process, that breaking off, like Dr. Fry said, that phosphate group. Myosin head grabs onto actin, pulls on it, muscle fiber shortens, basically.
exactly. And so on this slide, we have a stain for myosin ATPase. Now, not all myosin ATPase stains are the same. So anyone who's reading the scientific literature, you have to look really carefully to see, okay, whose method did you use? Because way back in the 50s, people started developing different methods. They're fairly, they're somewhat related, but they're not identical.
Mm-hmm.
And so when you compare results from different studies, you have to keep that in mind. What has evolved is most of the human work now, if you're doing myosin ATPase stains, uses the procedures of Brook and Kaiser. And that's been modified over the years by work from Dirk Pette and Robert Starren for human muscle tissue. And essentially on this one, it says there's an acid pre-incubation. What they found out over the years is if you take that little slice and you put it in an acidic solution, it'll knock out some of the myosin ATPase. Or if you put it in an alkaline solution, it'll knock out others. And so then you're staining to see what's left. And so in this case, with this acidic pre-incubation, you see that the type 1s are what's left. And because this is rat, there's not very much type 1. Humans, I can just tell looking at that there's hardly any type 1. That's often referred to as slow twitch.
Yeah.
But this is in rodents and other animals, there are muscles that are like have very, very little slow twitch fibers. Humans tend to be a much more of an even mix.
Yeah, and we'll get into more. I know Dr. Fry will talk about in a bit the characteristics of a slow twitch fiber, a type 1, versus a fast twitch fiber, the type 2s. But there are certain muscles we know, like in humans. For instance, the masseter muscle, it's a jaw muscle. You might wonder, how can you just chew gum all day long and never get tired? Well, the masseter I've seen in different studies is up to about 70% slow twitch fibers, and those are very fatigue resistant. So they can contract over and over again, and they just don't wear out. And so one of your jaw muscles that you use to chew is, from what I've seen, about 70% slow twitch.
And that's
do you
why,
do a lot of chewing?
That's why you can
do a lot of eating.
You can eat potato chips all day
yes.
long.
Oh, I can destroy a bag of potato chips. That is for sure. So that masseter gets a lot of work.
So here's a question: who volunteers to give a masseter sample? Give me that needle over there.
Right, here we go.
And are you going?
I'll lay down. I'll be a good boy and give you a sample.
But you can get into the literature and see that there are people have fiber typed pretty much every muscle in the body.
Yeah.
And that's because this myosin ATPase is viable for 24 to 48 hours after death. So during I suppose potentially autopsies, but at medical schools and stuff where they have a cadaver lab,
yep.
They're able to get a sample from some of these muscles that you can't access with living subjects.
Well, and a lot of the postural muscles are type 1 slow twitch, because the last thing you want is those postural muscles getting tired. You can't stand up, you can't walk around, like the soleus, which is underneath the gastrocnemius. It's part of your calf. That soleus tends to be, in most people, more slow twitch, because you need that to walk around and
So let's look at another
Yeah.
myosin ATPase. So this is the same sample as just the next slice. And in this case, it's an alkaline pre-incubation. And so the light fibers are the type 1 or slow twitch. And now the dark fibers are either are one of the type 2s, which are often called fast twitch. And I'll point out at this time, rodent tissue and a lot of the animal tissues and a lot of the animal research, they have 4 different major fiber types. Humans have 3 major fiber types in mature muscle. When I say mature, I mean during infancy, prenatal, postnatal, that doesn't count. If you have damaged muscle, injured muscle, sometimes you have recuperating, regenerating muscle tissue, that doesn't count. But in healthy, mature humans, you have 3 kinds. Type 1, type 2, of which there's 2 kinds in humans, 2A and 2X. In rodents, you actually have type 1, type 2A, type 2X, and type 2B. And one thing to keep in mind, different ways of staining use different names. So you'll hear fast twitch, slow twitch. You'll hear about red and white. You'll hear fast oxidative, slow oxidative, fast oxidative glycolytic, fast glycolytic. I mean, there's all these different names, but it's based on how they're stained. And they're not exactly identical because they're measuring different things. We'll see some examples here in a minute.
Well, I know with Thanksgiving coming up, I'm going to be focused on eating a lot of dark meat. I prefer the slow twitch type 1 fibers over the white type 2s. So
there's some truth to that, yeah. And just as a little aside, I'm not an animal physiologist, but those of you who are hunters and hunt for especially the migrating fowl,
yeah,
their breast muscles to be able to fly the long distances. These birds will go into training in anticipation of a long flight and working those pectoral muscles, which are important, and they're designed for the long haul.
Yeah.
Versus domesticated animals, where quite often you don't get the dark meat and the light meat as much because they don't migrate and and it's kind of bred out of them.
Yeah, that's interesting. So, want to go to the next one, the next
Yeah.
image?
So here's a different way to do fiber typing. So up till now, we've been talking about trying to determine which enzyme is in the muscle. Now we're looking for an important muscle that's part of the cross bridge. That's part of the head of the myosin molecule that binds to the actin.
Right. One of the proteins that allows that muscle fiber to shorten and contract. Yeah,
exactly.
Myosin.
And so each cross bridge actually has 2 what's called S1 units. And this is where the myosin heavy chain is. And myosin heavy chain in humans comes in a type 1, a 2A, and a 2X isoform, meaning a form of the same protein, but different forms of the same protein.
Like you said, remember Dr. Fry said humans have those 3 muscle fiber types.
And if this was a rodent, we'd be looking for a 2B as well. Right. People are able to, you can now buy antibodies that are designed to bind to specific isoforms. So on here we see fast myosin heavy chain antibodies that actually on the cross section, and it's the same cross section we've been looking at on the myosin ATPase stains, but the darker ones are the fast myosin heavy chains. Now, myosin heavy chain 1 is often and probably most of the time associated with myosin ATPase type 1, but they are not identical. And so you have to keep that in mind when you're comparing the results from a myosin ATPase stain with, they call this immunohistochemistry. And it's a very good method. Each method has its strengths and its weaknesses.
Yeah, and so with the immunohistochemistry, because I know I did some of it at KU Medical Center, but I was looking at brain tissue. But yeah, you're using, and I haven't done this exact stain for myosin before, but you're basically using an antibody, and that antibody wants to bind to an antigen. In this case, it's a protein, the myosin heavy chain, and then you can use a primary and a secondary antibody, and that can be labeled and kind of, you see it. You can see, what protein those antibodies are binding to, and then you can visualize it in the tissue section. So,
and we'll see a slide here in a few minutes. One of the challenges with the antibodies is some of them will also react with maybe another protein. So you get some cross reactivity. If you know that, okay, you can take that into account, but that means it doesn't just identify one, it maybe binds a little bit to something else.
Are they
and
polyclonal, monoclonal? The different types of antibodies. And you try to block that, and you try to, but this is again where Dr. Fry's right about this is if you're a student, if you're just someone in the general public that's reading through scientific literature and they start talking about muscle fiber types, I know you drive this point home. It's like, read through the methods, because a lot of people skip the methods. They read the abstract. They might go and find the purpose and hypothesis. They skip to the conclusion. What's the bottom line? That's great. You need all that. But that bottom line may be wrong if the methods they were using are different, or not done correctly, or how they're interpreting things is off because of the methods they used. And like you said, this is a complex area when we get into, and so many people write about this science, even people that aren't doing muscle fiber typing, they'll write like they're an expert on it. You actually are. You've done this for years. But I know it just grinds your gears when people start writing about, oh, this is a type 1, this is a type 2A or a type 2X. And it's like, well, no, that's not a type 2B. It's not in humans. Humans don't have 2B, so it just grinds your gears, I know. And any comments on that with some of the misconceptions and the issues with fiber typing?
And this applies to every field, and to myself as well. Know what your lane is, and know when you're outside your lane. So if we're doing some work, in fact, I just got off of Zoom before we came to film this episode, and it's in an area that I'm kind of outside of my lane.
Right.
So that means my colleagues need to be where they are knowledgeable in this area and they have experience, and it's just something to be aware of as you read the literature. Who's doing the research and writing about it, and then are the people that are interpreting it for the broader use. You have to read. Sometimes you have to read it carefully, and it can be really good and helpful, and in some cases it can be a little bit misleading. But
yeah, because the average person, you think of a strength coach, a trainer, somebody working in rehab, whoever it is in the field of sports science, that we don't want them being misinformed. I mean, that's not the goal of science in these studies. So it's, read through the methods, and it's important to be knowledgeable. And that's what Dr. Fry's trying to do here, is explain these different methods for identifying fiber types. So
let's look
go
at the next one. Yeah. This is another method of staining, and this just happens to be a stain for a particular enzyme. SDH stands for succinic dehydrogenase, which is an oxidative enzyme. So if your muscle cell is able to use aerobic metabolism very effectively, it'll have very high amounts of this. So you can
it's
see
a mitochondrial enzyme. It's in that powerhouse of the cell that really cranks out ATP, but it needs the help of oxygen in there. But part of the Krebs cycle in the mitochondria.
Yeah, and any enzyme, well, I shouldn't say any, but most enzymes that are in the muscle cell, they've developed a stain for. And so this is not identifying a type 1 versus type 2, because that's based on either the heavy chains or it's based on the myosin ATPase. This is looking for oxidative capabilities.
Right.
So quite a few of the type 1, the slower twitch fibers, will have very high SDH activity, but that's no guarantee.
That's a fatigue-resistant fiber. Yeah.
Exactly. And for example, you may say, you may get a sample from someone who's very sedentary, not fit at all, and you measure their type 1 fibers, their slow twitch fibers. They won't have a lot of SDH because they're not fit, and But at the other
They
hand,
don't do a
if
lot of running, a lot of
yeah,
cycling, things like that.
so even though they have a lot of myosin ATPase type 1 or type 1 myosin heavy chain, they may not have very high levels of SDH, or they'd have
Right.
less than what they could. Versus if you got samples from a marathoner, you'll see a lot of type 1 fibers, and that kind of a
Yeah.
stain will really take off. So the message here is different types of stains have different classification systems and have different names, and they don't all line up perfectly. There is a lot of overlap. Type 1s tend to have a lot of SDH in it, but it's something to keep in mind as you interpret the data.
Right. You have to think about the person that's in front of you. Are they sedentary? Are they a runner? What's their, because all of these are training adaptations. You know, if you're a runner, if you're a cyclist, you know, road cyclist, you do a lot of endurance activity. As an adaptation to that type of training you do, there's going to be a lot more of this succinate dehydrogenase enzyme that's in those type 1 fibers, because you're relying heavily on that enzyme to help you with the production of energy to fuel that type of training, the type of racing you do. But like you said, you have to be careful because, well, somebody that's sedentary, they may not have a lot of that enzyme, but if you do a different stain, you might say, oh, well, they have a lot of type 1 fibers, you
Yeah.
know? So that's where it's, the interpretation is so key, like
Yeah.
you mentioned. And you want to go to the next
Yeah.
next slide, slide 4.
So here is a photomicrograph out of our lab, which is a human muscle biopsy. And you can see this is actually an acidic stain. It's pH 4.6. And the nice thing about this one is this kind of pre-incubation really brings out the most different muscle fiber types. So the work coming out of Dirk Pette's lab, he was from Constance, Germany, and Bob Sterin at Ohio University, they were able to differentiate up to 7 and potentially even more fiber types. So the 3 major types, type 1, type 2A, type 2X, but then you have fibers that are kind of a combination of the two. And so
Hybrids.
they're called hybrids. And there's some important implications for that. In this one here, the black fibers are type 1. The really light white ones are type 2A. And then the dark gray are 2Xs. And then they have a lighter gray, and those are 2AXs. So it's a little bit of type 2A, a little bit of type 2X. And what's just kind of interesting to notice there, it looks like kind of a crack along the right side of that image. That's the border of a fascicle. So if you study the connective tissue layering of the muscle, the epimysium, paramysium, endomysium, that's your fibers are kind of put into groupings.
They're bundled.
And we just happen to catch one there. But we can see the cross-sectional area of these fibers, and this is just a nice stain showing very clearly the different fiber types. If we go to the next slide, this was actually from the cover of the Journal of Applied Physiology back in 1994, out of Bob Sterin's lab, and it shows to be able to determine all the subtypes using Myosin ATPase, you have to use all 3 pre-incubations. So you have a section that is in the alkaline, pH 10.4, and then you have 2 sections that are in either the 4.3 acidic or slightly less acidic 4.6. The sensitivity of that enzyme is that tight, where just a little bit of difference will knock out some and not others.
Yeah.
And if you put all 3 of those together, then you're able to correctly classify up to 7 fiber types. And it kind of depends what the purpose of your study is, because you don't necessarily have to do all of those stains, but the most precision occurs when you do that. And that means you may have 1,000 fibers, and that means you got to stain them all, and that means you need to count which ones are which, and separate out the light gray from the dark gray, and is it black or white on the other section, and we have to trace them.
Yeah.
And so this is not fast work.
Yeah. Oh, yeah. Like a lot of things with research, it can be very tedious and time consuming. But it's always worth it in the end when you find something new and interesting. You want to go to the slide 5 now?
Yeah.
Slide 5.
So here's immunohistochem. We talked about the different antibodies. And what's interesting is a primary antibody is what you get from, there's supply houses for these purposes that create these antibodies specifically for the different myosin heavy chains. On the left, we see an antibody, it happens to be a F8, which is looking for the type 1 myosin heavy chain. And so that primary antibody gets put onto that slide, and it's looking to bind to that. And in the middle, we have a BF35, I believe it is. That is looking primarily for type 2A, but it also picks up a little bit of type 1. So you can see the really green versus the slightly green. And then the one on the right is an antibody that's just looking for the 2X's. And then once that antibody's on, you have to put a second antibody. That's a secondary antibody. And that has a label that you can see. In this case, it's an infrared label that shows up a different color. So you have to have an infrared camera. And that's why you see the blue, the green, and the red. It looks slick. And on a good stain, it really shows up like crazy.
Yeah, that secondary antibody is like flipping the Christmas tree lights on, right? You just see the whole tree. It's like it lights everything up.
Yeah,
basically.
yeah. And so then if you go to the next slide.
Or next, the arrow down here. This shows the merge.
So we can put all three of those together. And so you can see, okay, it's very clear which are the 2A's. We can see the 2X's. We can see the 1's. The blue doesn't show up real well on that, but, you can put different filters on. One of the challenges with this is the hybrid fibers. We know it's a hybrid. We don't know how much. Is it a lot of A and a little X? Or is it a little A and a lot of X? Or is it about a half and half? Where some of the other methods that we'll take a look at can give us a little more precision.
And we'll talk more in a bit about that. But, the types of training you do, the types of exercise, there can be some fiber type shifting. And, a fiber that's maybe a 2X may start to become more like a 2A or become more like a hybrid that's in between. So it's really interesting how the types of training and the exercise that you do affect this. And of course, some of it's, genetics, which we'll talk about. You can't pick your parents, but, there's always a genetic component to things. You want to go to slide 6 now?
Yep.
Slide 6.
So here's a different approach. You remember I said the cross bridge has this big protein, myosin heavy chain. It's one of the bigger proteins in the muscle. If you get a sample, you can either section it off into little slices, you can get a bunch of those, or you can just get a chunk of the frozen sample and grind it up. But you basically break it down and you lyse the sample, which means you get the proteins to pull apart. You don't want to destroy the proteins, but you want to disassemble them. And so then you use a procedure called gel electrophoresis, which now you take that lice sample, you run it through it's sort of like a jello-like substance, but it's because it's flimsy, flexible, but you run a current through that and it pulls the proteins down through it and it separates them based on their molecular weight, their size.
They're literally running in lanes like athletes in a sprint would run in different lanes on a track.
Exactly.
Yeah, you run these proteins in different lanes.
And so here is a human
and I should say the lighter weight proteins run further than the heavier weight proteins. That's an important point to remember too.
So it separates them based on their size.
Based on their weight, their molecular weight, yeah.
And so here we see the result of one individual who we ground their biopsy up, put it through this gel electrophoresis, and then we stained it with a compound called Kumasi blue. And just a little side note, a lot of these stains that we use for biological purposes, they come from the textile industry. They're clothing dyes. And if you look up where did Kumasi come from, it's a dye that has been used in the past, and that's the case on some of the other dyes that we use. But it does very well because it's looking for a protein. It doesn't care what protein, it finds a protein. And so we look at these 3 lines where we've got good separation, And it's really hard to get separation between the 2A and the 2X. That's the biggest challenge. But if I look at and I scan that image that we see, I can see how much area and how dark is each band. And that tells me in that biopsy what percentage of that muscle has type 1, what percentage has 2A, what percentage has 2X. And it does not fiber type. Because think about this. Before we took a section and we stained and we could see each individual fiber, either for the myosin ATPase, for an enzyme, or we took an antibody and put it on it. This, we ground it all up. So we have it's a stew. You're making stew on the kitchen stove. Everything goes in, you mix it up, and you eat it all together. Well, in this case, we took all the sample, ground it up together. And so this is really related. The amount of band is associated with the cross-sectional area of that muscle sample that is a type 1 or 2A or 2X. So it's not fiber typing, but it does give you some really important information about that sample.
And you look in this sample, I mean, look how thick the type 1 myosin heavy chain is there. I mean,
so that's probably 50 to 60% type 1 is my guesstimation, but we can get a very precise amount when we scan it.
Yeah, yeah. And then I think go ahead and click. So here we're looking at the rabbit, the gastrocnemius muscle, so the calf muscle with the rabbit.
So I put this in here because it's been well documented. You know, we've talked about some of these animals, they have more fiber types. Well, here we see that there's actually 4 myosin heavy chains. So when you interpret the animal literature, sometimes it has application to human data, but sometimes maybe less so. And you can't, the fiber types and the myosin heavy chain isoforms are not identical.
And this is where sometimes it gets messy with the way things are written in articles online that you may read, or sometimes even in textbooks, which it shouldn't be. But then I guess it's who's writing the textbook, right? Who's editing it? Are they an expert on muscle fiber typing or not? Or do they just think they are?
And that's true. And a number of years ago, the names of the fiber types changed. Because originally, if you read some of the older literature, and this adds to the confusion, humans had 3 kinds: type 1, type 2A, type 2B.
But now we call that 2X.
But we call that 2X because it was found that the human 2B was closer to the animal 2X. Now, when I say closer, is like that's absolutely true. They come off, they have some genetic similar sources, but they're not identical. And however, just know if you're reading the older literature and they're talking about humans 2B, they're probably talking about what we now call 2X. However, in the animal literature, and in this example, we have A, X, and B. One thing I'll point out is if the 2X in animal is the same as the 2X in humans, we got a little problem here, because notice on both the human and the rabbit in this case, the smallest myosin heavy chain is type 1. It goes the furthest.
Right. It's at the bottom.
But look where 2X is. In the rabbit, it's lighter or smaller than the 2A. But in the human, it's the other way.
Because it's at the top.
It's at the top. These are not identical. And so, we could, it's kind of beating a dead horse as to why the names were changed. Suffice it to say that now we call the human the fastest isoform 2X's. But again, it's not identical when you compare animals with humans.
And then we'll go ahead and click. So basically, kind of your comment here, this is not fiber typing. Maybe go into some detail there about that.
And when you read some, in the last 5 or 10 years, there have been some reviews that have come out that are talking about changes between type 1 and type 2 muscle fibers, and they cite all these studies. The problem is they're citing myosin heavy chain data, which is not fiber typing based on doing it this
Right.
way. If they were doing immunohistochemistry, okay,
Staining. Yeah.
then that would be valid
Yeah.
where they're looking at individual fibers.
Yeah.
But we can say for sure that the proportions of these different protein isoforms are related to the cross-sectional area of that muscle that are the different fiber types. So it is important information and just part of the total picture.
It's just this is where words matter. How you say things and how things are written is important
Yep.
to be expressed in the proper way, right?
Yep.
So it doesn't lead to confusion.
Yep.
So we go to slide 7.
Yeah. So another way to analyze muscle is you take a biopsy, and remember I said you may have, on a good one, you may have 1,000 fibers.
Mm-hmm.
Let's tease out the individual fibers.
Which is what you see here.
And this comes out of Dr. Scott Trappy's lab at Ball State, where for years they've done this kind of research. And this has been done, in a number of labs for a long time. But I had an opportunity a number of years ago to visit their lab. This shows a single muscle fiber that's tied off at both ends. And they'll do a number of interesting measurements, including looking at the force it can produce and how quickly it contracts, as well as the size and the fiber types. So if we go to the next click, they'll take each one of these individual fibers after measuring performance. It's like you and I go in the weight room. Let's see how my bench is today. I'll do a 1 rep max, or I'll do reps to failure, or I'll look at lifting velocity. They do that with the individual fiber.
Right.
But then once they're done with that testing, they'll run it through a gel, and each one of those lanes here is a single fiber. And you can see that they can say, okay, definitively, this was a type 1, or it was a 2X, or it was a 2A. And this is from humans, or it's a hybrid fiber, has a little bit of A and a little bit of X. And so this is another way to fiber type. The challenge here is how many of these single fibers can you get? Because this is really a fantastic capability, but the numbers that you can get is limited.
Mm-hmm.
And so even the labs that are very aggressive in this, the numbers that they analyze are quite a bit less.
Yeah, and obviously you need enough fibers for the study to be adequately powered. You need a large enough sample size. But yeah, that's really, really cool.
Now on the next click, we'll see another variation of this. If you take a single fiber, no, let me back up. If you take a section of tissue that's now mounted on a microscope slide, like we talked about earlier, you freeze dry it, and then you tease out the individual fiber that's only 12 microns thick. So you're comparing that with the stained, you got some other slides that you've stained. And so this is also single fiber, only you don't have a full length of muscle. You can't measure its forces or anything, but you can look right there. And so you see these stains here, which are myosin ATPase stains. And people will, some of the people in the literature say myosin ATPase has not been validated against the myosin heavy chain. And it's like, well, that's wrong, because, over a third of a century ago, or about that long ago, this has been done. It's been out there forever. They have actually shown that the myosin ATPase stains agree very closely with the heavy chain expression, and that came out in 1991.
Know your history as a researcher, right? Go back and look at the older studies that have led up to the development of the different methods and where we are today.
Yep.
Yeah.
Let's see what else we have here.
So we'll go on to slide 8. Now on slide 8, here we go. So can muscle fiber types change, right? So this is a question I get, I'm sure you get all the time from students too. I mean, people want to know this. Well, there's a genetic component, right? I'm sure if you're the son or daughter of an elite Olympic sprinter, that maybe 80% type 2 fast twitch fibers can sprint down the track at crazy speeds and is probably very strong and their fibers produce a lot of force. You may more than likely have a lot of fast twitch fibers because of that genetic component. But with training and can these fiber types change?
Well, let's take a look at this. let's look at the next click. And this comes from, in my mind, a classic study that came out of Bob Stearns' lab, where they actually took untrained people. So now that's an important thing to remember. These are not already trained athletes. These are sedentary, untrained people. Why that group? Because at that time we wanted to see, okay, will we get the adaptations we think we will? And so we set the stage with which subjects do we think would show the most adaptations? Probably people that are untrained. Now, that has some implications. Does that translate to training elite level athletes that have been training for 1 or 2 decades? That's another story.
Right.
But what we see here are some myosin ATPase stains. And on the left side, we have a control subject who, this was around 6 weeks of strength training. No, 8 weeks of strength training. And the control pre was the fiber type from the control subject who did no weight training. And then 8 weeks later, we got another biopsy. And actually, they did biopsies before the study, after 2 weeks, 4 weeks, 6 weeks, and after 8 weeks. So they could see the progression going through this strength training for the lower body. And if you look at the control data, both pre and post, we see that there's not a lot difference in the fiber size. And we still see, to me, the telltale, and in this one, you notice that it's labeled 2B. We still see 2B fibers. I kind of like what Bob Stearns used to call the 2B fibers. He called them couch potato fibers. If you're very sedentary, you probably have a bunch of those. And as soon as you start doing, and especially with lifting, they'll start disappearing. But in the control subject, they didn't. The fibers didn't get bigger. We still had the fibers that are prevalent in many sedentary people. On the right side, we see in a pre and a post sample for a weight trained person. And just the visual appearance
So just
from
to
pre
make
to
sure I got
post.
this correctly on this study, the control pre and control post, like you said, these were sedentary individuals. You got the sample, the control pre, and then they did 8 weeks of resistance training.
Nothing. They did nothing.
Okay, that's where I was confused. I thought you said that. So they did nothing. Okay.
So they did nothing. On the right side, the weight trained, they did the training program. And you can see the fibers are larger.
But they were not trained weight. They hadn't been doing any weight training coming into the study.
Correct.
So they were sedentary. You got your sample pre, then they did the 8 weeks of training, and you got your weight training post.
Exactly.
Okay, I got it now. Okay.
And so on this, you see in this example, there's quite a few of the 2B or now called 2X fibers. Those are almost completely gone. Or I mean, well, that picture, they're almost all gone. I guess we do see a little bit, but they really have decreased. We see a lot more of the 2As.
Right.
So what do we see? We see fiber growth, hypertrophy. We also see a fiber transition from the 2Xs to the 2As. And in the weight room, that's a very, we see that a lot of the time.
And again, when he says 2Xs, those are labeled as B here, but we call them 2Xs in human. This is human tissue. So there's been a fiber type shift or transition from the 2Bs, which we would now call 2X in human, to a bunch of 2As on the right side. And like you said, they're bigger fibers too, more type 2A.
Yeah.
And we'll talk more about what that means. Well, what's the difference in the structural and functional characteristics of a 2A fiber versus the 2B or 2X or versus a type 1?
So what we find out, as soon as you start, in the weight room, you can lift heavy and you can recruit those fibers that are, we call them high threshold motor units, hard to recruit fibers, but you can do that in the weight room.
Right.
And get them activated, and they begin a conversion to 2As.
And what percentage of 1 rep max are we talking here?
Well, this was
to really get that conversion to happen.
This was a fairly generic program. It was leg press, leg extensions, and leg curls, and Smith rack squats. And
so this is section from the vastus lateralis.
Vastus lateralis. Trained 3 days a week for 8 weeks. They did 8 to 10 RM. Let me get this correct. They did, I believe it was 6 to 8 RM one day, 8 to 10 another, and 10 to 12 on another day. So they varied a little bit, but it was as heavy as they could handle.
So they're at least up around 80% of their 1 rep max, somewhere in there. I'm just kind of spitballing that in my head, but it's moderate to heavy training. It's not light loads. I mean, we're not talking doing, sets of 30% of your 1 RM. Yeah.
And definitely not. And getting these folks to complete this was, this was pushing them pretty hard. We got, it was really good results. One thing to also point out, one of the lifts was a leg press, the 45-degree angled where you lay down on your back and you lifted. The number of reps you can do at percentages of 1 RM are quite different on that.
Oh, yeah.
Compared to a squat or a bench press. And so you can do, so that was
take a biomechanics class with me or Dr. Herder or somebody here, and you'll understand why, right?
Yeah.
Trig.
So this is not sets of 30. This is not, this was a controlled rest interval. On one hand, nothing outlandish. We gave them a little bit of a variety. It was not a long-term periodized. It was just 8 weeks. And
I think that's really interesting. It was only 8 weeks, and you see that fiber type shift. I mean, it's pretty.
And I'll tell you what's also interesting is these are images from a male, and we saw statistically significant changes occurring within 4 weeks. For the women, we saw it within 2 weeks. So within just a few training sessions, the transition begins. So the adaptation starts coming out of the gate. It took 2 weeks or 4 weeks before it was statistically significant for this population. If you were highly trained to begin with, that's another story because you're not starting from the beginning.
Right. Yeah, you're starting closer to your ceiling. So yeah, it's different. Now you said, what was the frequency? How many times were they training their legs again? Do you remember?
I said 3 times, but I think it was 2.
Two times a week.
I think it was 2. So because I remember part of the story is within 4 training sessions, we saw some of the changes were starting to be statistically significant after 4 sessions for women, after 8 for the men. But everyone was going in the same direction.
That's interesting. So yeah, remember that moving forward, there's this shift with weight training from the type 2X or 2B as shown here to the 2A. You want to go to slide 9?
Yeah.
We got this little, this is showing.
This just shows the actual percentages.
So we're still on slide 8 here. I'm keeping track of this for when the video gets edited. But we're still on slide 8 looking at the table.
This just actually shows some of the numbers for that these images go along with. So if you look towards the far right, one thing I'll point out, we've got a lot of fibers to count. On average, it was over 1,000 fibers.
Yeah, week 1 there's 1,156. Looks like week 3 1,331. Man, that's every week over 1,000 fibers.
And so that's important because if you're trying to get accurate numbers and you only have 100 fibers, and you've got how many different fiber types, there's more room for error. And so that's one strength of this. But also notice if you look down the next column, second from the right, it's the type 2B, which we know is really the 2Xs. We see that basically those decreased percentage-wise by half.
Oh, yeah. Because at week 1, it was 20.7, and then by week 9, it's down to 9.5 in that type 2B column.
And you begin, and even though it was not significant, you begin to see the 2ABs start to creep up because you're seeing a shift to where some fibers now have a little mixture. The two A's are starting to creep up. It's not statistically significant. The type 1 started at 40%. They ended at 40%.
Yeah.
And so that's just, it gives you some actual numbers as far as what to expect.
Yeah. Okay.
Now we can move on.
Now we'll go to slide 9 officially.
So here's a few things I do want to point out. So if we can go ahead and click, remember humans have 3 isoforms of myosin heavy chain. Animals have 4. And as you read the literature, keep that in mind. Next one. Now we have what used to be called 2B. And the capital 2B is the myosin ATPase designation. The small 2B is the myosin heavy chain. They're now called X's. So as you read the older literature and mix it with the new, just keep that in mind. The next one.
Do you know about what year that changed? Where what was called 2B in humans now is being referred to as 2X? I mean, so if I'm going through the literature and I'm like, okay, this is a study from 1995.
Well, it was probably 90s.
Okay.
Because Stern's work that I was closely associated with, we used some of this actually from a historical perspective. Two different labs made almost identical discoveries. It's kind of like the sliding filament theory. You had two Huxleys, Hugh Huxley in his lab, Andrew Huxley in his. They came to the same conclusion. They actually coordinated their simultaneously published papers back to back, but they were separate labs. And so you had Pete's lab that discovered what they called a new fiber and a new protein in animal muscle. They called it 2D. And at the same time, I think it may have been Schiaffino's lab discovered it, and they called it 2X. And why did Pete call it 2D? Because they found it in the diaphragm.
Oh, okay.
Well, they found out they were talking about the same protein. Okay, so let's make it simpler, and somehow the 2X designation won out. And so then you began to find out that Smerdu published some stuff saying, well, the animal 2X is closer to the human 2B, so there's like 80% homology, and the genetic origins are similar. So then that happened, and that was in the 90s. It wasn't like one set time.
Right. Okay. So around that time. Yep. And then I guess we're on our third bullet point here. So talking about the myosin heavy chain bands.
Yeah, there are some papers out there that actually they have a picture of the bands and they mislabel them. It's like so just keep that in mind. Yeah.
Next bullet point.
If you measure myosin ATPase staining, that is not measuring the heavy chain. That's measuring the enzyme. Are they closely associated? Yes, but you're not measuring the heavy chain.
Right.
And then underneath that, our next one, people say that the myosin ATPase histochemistry has not been validated with single fiber. In fact, click one more time, I think. It hasn't been validated with single fiber heavy chain content. It has for a third of a century. That's an incorrect statement.
Okay.
Our next statement here is you cannot get percent fiber types from percent myosin heavy chain content.
Right, and this is where, again, like you said, because you're making a stew, you're grinding up that tissue. Yeah. It's a homogenate.
If you are using antibodies, immunohistochemistry, you can, but from the ground up relative content of myosin heavy chain, that's not a fiber typing method, and that gets misinterpreted. Our next item is related to that. You can't do fiber type shifts, so if you're looking at transitions. And where am I going with this? Just imagine you have something called, and this occurs, and we saw images of it, preferential hypertrophy, which means I get growth of my 2A fibers, but I don't get growth, or I don't get as much growth of my type 1. So now I've got more type 2A. Let's assume there's no fiber type change, no conversions, but I now have done something, probably lifted. I got bigger type 2 fibers. That means I'm going to have more type 2A myosin heavy chain.
Right. In the homogenate.
But there's no fiber type shift.
Right. So you only know that if you do the staining, right?
Exactly. So, and that gets misinterpreted as,
that's a key point.
Type 1 switches to type 2 switches to type 1. We got it all over the place. It's like we don't, or we haven't measured correctly. It might be happening, but we don't have the right
Right.
measure.
Yeah, that's exactly right. And you would think, yes, lifting those type 2 fibers are going to get larger. And so you're going to pack literally through protein synthesis. There's going to be more myosin protein, heavy chains in there.
Yep.
So yeah, it's right to say there's a greater percentage of the myosin heavy chains, but how do we know a fiber type shift occurred?
Yeah,
Yeah.
exactly.
Yeah.
Exactly. And
Yeah.
and then what else do we have here? So the net result is some of the reports of shifts from 1 to 2 are incorrectly reported. Now, do I think you can switch from 1 to 2A and 2A to 1? Maybe. Those are hard studies because those are really long-term chronic adaptations. If I've been you see such extreme fiber types in some very highly trained elite athletes. Um, Maybe they were born that way. I don't know. We didn't test them. Maybe it's the result of a long, it's not a 1 semester, it's not a few months training block, it's years and years, and some cases decades of adaptation. Maybe so.
Yeah, so this would be an example of this is like, let's say you have a Kenyan distance runner. They genetically tend to have a lot of type 1 slow twitch fibers. They can run forever, not get tired. Those type 1 fibers are fatigue resistant. But then if you put them on a weight training and a sprint program, yeah, are they going to shift from a lot of those type 1 slow twitch fatigue resistant fibers to the more fast twitch type 2s? I mean, this has been debated, and you see it even written differently in textbooks, and you hear one thing from one person, another from another. So you think, you're speculating, you think there could be that conversion with that example?
Sure could be. One thing too to keep in mind, we use as examples someone like your Kenyan runner, an extreme endurance athlete. At the other end, we're looking at a weightlifter or a shot putter. Okay, that's the other extreme. Highly anaerobic, highly aerobic. Most people are somewhere in the middle. My sport was wrestling. Oh, I need endurance.
Right.
I need strength. I need power. Maybe I don't want muscle growth. It's a weight class sport.
Yeah.
But muscle's functional, so maybe I do.
Yeah, your average person is about 50-50, type 1 and type 2.
And so many of your sports are somewhere in between those extremes. And you need, and you also have to realize your 2A fibers, if I'm a shot putter, I can train them to be really helpful for my shot putting. If I'm a distance runner, I can make those highly oxidative. Those 2As, they're kind of a jack of all trades, depending on how you train. And so
and I think it's also important for people to remember, I mean, they see it here, the fiber type shifts from 1 to 2, but there's a whole spectrum of hybrids in between there.
Yes.
And we didn't even really get into that. I mean, a little bit, we mentioned how there's hybrid fibers in between the type 1 and the type 2A, and in between the 2A and the 2X. There's this whole spectrum of hybrid fiber types.
I think I may have one more thing here.
Yeah.
Nope, but don't.
So we're on to slide 10 now. And this is showing structural and functional characteristics of muscle fiber types. Now, this was from a study way back in 1967, and this was rat tissue. The reason I threw this in there when we were putting our slides together is I think just for the general population, an undergrad student, this helps to, I mean, it's very simple. Obviously, as Dr. Fry was talking, this is fiber typing stuff is very complex, but this is just very simple. I think it helps people understand, well, what are the differences between an ST, a slow twitch type 1 fiber, and how about a fast twitch A fiber? They're in the middle column. And then a fast twitch B, which remember it's B because in animals it's B, but in a human, we now call that fast twitch X. So maybe if you just talk about some of the different characteristics between the slow twitch type 1s and then the fast twitch type 2 fibers.
So as Dr. Taylor was mentioning, there's, besides just the fiber type, as we've talked about, these different categories have different characteristics: how much force they produce, how rapidly they contract, what is their fatigability, but also just structurally, how many fibers are in a motor unit. What is the size of the motor neuron? What are the, how much of a, how much of a sarcoplasmic reticulum is there in there? Conduction velocities. It does talk about the type of myosin ATPase. And as we were talking in preparation for this interview, the date 1967 is apparent because they didn't have the 4 fiber types for the rodents. They only had 3 here, and we'll see here in a minute that over the next decade or two, a lot got discovered and added to it. But there's a lot of unique characteristics and also a lot of overlap between these different categories.
Yeah. And I think one thing, like I'll just mention a few things. If you look at motor neuron size under characteristics, that second row there. So if you look at the 2 fast twitch fibers, they're innervated. Fast twitch fibers tend to be larger, and they have a larger motor neuron that connects to them. And what does that motor neuron do? It sends an action potential, a nerve impulse from the spinal cord out to those fast twitch fibers. And because the diameter of the motor neuron is very large, that nerve impulse gets there more rapidly than a smaller motor neuron size that you see with the slow twitch. So then this helps to allow those fast twitch fibers, because there's a faster nerve conduction velocity going to them, that impulse is getting there quicker. They contract earlier, they contract with more speed. And you see that if you look at the contraction speed as well in milliseconds there, 50 milliseconds versus maybe 110 for the slow twitch.
Yeah, and you bring up a good point. You notice on this slide, and we'll see it on the next one as well, it says, what's the force that can be produced by the different fiber types? And you notice it says, and that's third from the bottom there, the slow twitch, it's low force, and the faster categories is high. What we have found out is a lot of that, in fact, pretty much the majority of that difference is because the fast fibers are bigger. And so just the ability to produce force is highly dependent on size, which is ironic because there are several authors who recently have said, oh, size doesn't matter. And it's like, well, size can you be stronger without increased size? Sure. Anyone who's studied all the many ways your body can control force should know that. But in this case, at the muscle fiber level, bigger fiber, more force. The difference, though, how fast it contracts. So now force times velocity, the power differences are huge.
Right. So you got those fast twitch fibers tend to contract, get that impulse, nerve impulse to them. It arrives more quickly. They contract more rapidly. So they shorten quicker. So there's your velocity component. And then if they tend to be larger fibers. If you have a lot of fast twitch fibers, maybe genetically you have a lot of fast twitch, and then you go do a bunch of weight training, you're going to get pretty big. And if you have a larger fiber, there's going to be more protein packed inside that, more myosin, more actin filaments, more of these what are called sarcomeres that allow you to produce force when that muscle fiber shortens. And so more protein packed in there, more sarcomeres, more contractile elements, contractile proteins, you should be able to produce more force. I mean, it only makes sense that there's pretty direct correlation between a muscle size and its ability to produce force. Now, if we look at slow twitch, one of the great things about slow twitch fibers is, if you're an endurance athlete, you're going to love these and want to have a bunch of them because they have a great aerobic capacity. They're able to utilize oxygen very well. They have a lot of mitochondria, great capillary density. It's easy to get blood flow and nutrients into them. And those things can just contract and contract and contract, and they just are very fatigue resistant. So you can run and run and run. If you got a lot of slow twitch fibers, you're probably going to prefer those sports like, running a marathon or, a road cyclist, any type of endurance, cross country skiing, so because those are very fatigue resistant fibers. They don't produce as much force as the fast twitch, but man, you can really, really use them. They have a great aerobic capacity.
And you have to realize too, this table is from rats. So like when they're showing the fast twitch bees on the far right, that's not the same as the 2X's in humans. And because an endurance athlete, a human endurance athlete is not dependent on 2Xs. In
For
fact,
sure. Yeah.
those aren't very good at
Right.
glycolytic or anaerobic. They're like we say,
Right.
they're kind of couch potato.
Yeah. But yeah, those are just anything else you want to point out? Differences between slow and fast twitch?
Let's go to the next slide, which actually I think, uh, now I'm not expecting anyone to really be able to decipher all this, but this comes from about 2 decades later. And this is from Sato out of Japan, where it was a nice summary of the mountain of research that was coming out at the time. During those 2 decades, the nice thing is this identifies, one, it's on human data. So now researchers are getting access to human muscle. They are doing these tests. They're looking at many, many variables. The ones that had the red arrows are related to speed of contraction, the force capabilities, the twitch, tension, and factors like that. And so a performance perspective. And it's even, you can go to other sources and see where it's even more up to date.
So I know the one on the far left, looking at this table, so this is slide 11. That first arrow is myosin ATPase activity. And so you see it low in the slow twitch and high in the 2 fast twitch fibers.
So I would, if I was revising this slide, I would say that the difference between, if you were to label those at the top, type 1, type 2A, and type 2X, I would say it's not so much a measure of activity, it's the speed at which that enzyme works.
Right.
So how quickly does it break down that ATP molecule? And so the slow twitch, the type 1 myosin ATPase, does it just a little more slowly. And the type 2A, faster. The type 2X, even a smidge faster. The problem is in humans, the type 2X, you start using it at all, it has to convert.
Yeah.
Unless you, now, there are some reports of some elite athletes that have quite a bit of type 2X. You have to go back and see how they measured it. But also, when you get to some levels, is it through just natural selection, your genetic makeup? Is it through some types of training that are conducive to it? And so that's still an active area of research.
Right. And, that's important, the myosin ATPase activity. If you think about it, if it's in a fast twitch fiber and you can split off that last phosphate group from an ATP molecule with the help of this enzyme at a more rapid rate, that's going to allow the myosin head to reach out and grab actin and shorten that muscle fiber. And if that's accelerated because this ATPase enzyme works at a more rapid rate in fast twitch, you can see how that's going to improve your power output. I mean,
exactly.
So really interesting how just the difference in the enzyme activity, ATPase, that allows you to use that fuel, ATP, for muscle contract works more rapidly in the fast twitch. And then I guess the next arrow there, Andy, Dr. Fry, if you want to talk, is speed of contraction between the slow twitch and fast twitch fibers.
Yeah, and on this table, they don't really differentiate between the 2As and the 2Xs, but there is a difference. And that's been shown at the single fiber level. It's been shown in the muscles that are predominantly one fiber type or another. And so that's related to the enzyme. That's related to other proteins. I don't even want to get into detail on this, but on the cross bridge are also proteins called myosin light chains. And they, which myosin light chains are there can impact the speed of the velocity of that cross bridge action, and they can get modified. They can be phosphorylated. And that's one of the questions, where does that fit in to muscle performance? And we actually have, right now, we have a visiting professor with us, Dr. Lauren Chu from University of Alberta. And he's not the only one. There's been several that have visited my lab in the past, but he's published stuff looking at the light chains and how they impact performance and maybe are contributing. In his case, he was a national champion weightlifter for Canada a number of years ago. And so he was studying what's the role in actual weightlifting performance with highly trained weightlifters.
Right.
Is it the only thing? No, but it's certainly a factor, and that would at least conceptually contribute to speed of contraction.
Yeah. Okay. And then I guess the next arrow, if we keep going down, is twitch tension. And you see it's marked low for slow twitch, for the fast twitch A, intermediate, and for the fast twitch B, which we now call fast twitch X in humans, high.
So this isn't an isolated preparation. You're not working through the skeletal system or anything, and they're just measuring how quickly you stimulate it and how quick can it move. Those two X's can be, they're the fastest. It's just if you start using them very often, they're not, they don't stick around very long. Theoretically, if I'm a speed athlete, I'd like to have a lot of 2Xs, except their endurance is pathetic.
Right.
And so it's like, well, what's my event? And you have to realize when we're talking fast and slow, this isn't like smooth muscle in your gut or something. This is still measured in milliseconds, these speeds of contraction. So even
Right.
your slow twitch, you still can move.
They're still contracting in milliseconds.
Yeah,
it's just a little bit longer
yes,
the
exactly.
time than the fast twitch. So yeah.
Let's take a look at the next slide, maybe.
Slide 12. We'll move on to that.
So this is a neat paper. Woodrick, these were taken from his table. So these images were not from his paper, but the data is. And it just basically shows different single fiber characteristics. So you've got across the bottom, is it myosin? What's the myosin heavy chain content?
Yeah.
So you have type 1, you have 2A, you have 2X, and then you have hybrids that are kind of a mixture. Well, how much force is produced? And a lot of times that's related to the size of the fiber. You see that so on the top right, there's a slight difference in the cross-sectional area of the fibers, and that increased size means there's more heavy chain that can contribute to that peak force. And then
you see the type 2s, the 2X is the largest.
Yep.
And then you see the 2As and the hybrid 2A, 2X. All of if it has some type 2, you see if it's a type 2, larger cross-sectional area, basically.
And this is also one where you have to go back and look at the methods because, for example, in the workout of Starin's lab, 2Xs were never very big, never very big. And so, are these untrained? Are they, of course, if it's animal stuff, well, that's completely different.
Mm-hmm. But you do stay with the 2As. I mean, if you look at 2As, pretty much all studies are going to agree on that. The 2As are larger than the type 1.
Oh, yeah. And I'll tell you, the athletes that, well, if you're talking about an endurance runner, they don't need size.
Right. You don't want it.
They want smaller, so they, the weight they carry. Also, the diffusion distance from the capillary to the metabolic machinery in the cell, I want that to be really close. I don't want a great big cell I got to get.
Yeah.
And so it becomes more efficient.
You don't want a large cell for all the nutrients, oxygen, everything to have to navigate to get into the.
Yeah. One group of athletes, so that the slow twitch, the type 1s, seem to grow almost as much as the type 2s are bodybuilders.
Yep.
And because they're not worried about performance from how much did I lift, how fast did I run, how far did I throw the implement. They want size. Mm-hmm. And no matter who you are, a lot of your fibers are going to be type 1s. Well, I want them big too. And so I think that's a reflection of why many of the training strategies they adopt, where they are tapping into that pool of fibers, whereas other athletes are like, sometimes, in fact, arguably, do the type 1s slow down? They're kind of like dragging a weight around because they contract more slowly. I want the fast ones to be doing all the work. But you can still see the power is related to fiber type. The shortening velocity is related. What this means in an intact human with everything else functioning is another question.
Yeah, so basically those asterisks are just indicating significant differences. So that the type 2s are larger in size, produce more peak force, greater peak power, and the velocity of shortening, how rapidly they shorten and contract, is also higher in those type 2s compared to the type 1s. But the major benefit, like we've talked about and hammered home with the type 1s, is it's just they're very fatigue resistant. And so as an aerobic endurance athlete, that's a plus for the types of events that you're competing in.
Another thing, the last thing I'll point out on this figure is this is where you're artificially coming in and stimulating these fibers to contract. I know, or whoever the researcher is knows, I'll get them to contract. If these are in your or my leg, I've got to be able to recruit them, which is another story. Can I recruit? What do I need to do to voluntarily recruit these fibers maximally to get them to activate and contract at these speeds? And it's a function of the individual, of their training methods, and it's related to the sport they're training for.
Yeah, and generally, and this is, I guess it's one of my questions, but leading into it, like with aging and inactivity, you see generally as individuals get older, you start 60s, 70s, 80s, 90s, you're inactive, you're not doing any weight training, you're not doing any explosive type movements, sprinting, jumping, things like that. You tend to have a loss of those type 2 fibers.
You use it or lose it.
Because those are the fibers that, as you see here, you really recruit to produce a lot of force and a lot of power and to move quickly and explosively. So, it's like I used to recommend to older individuals that I was training before I went and got a Ph.D. and was training clients. It's like we still need to maintain some of these type 2 fibers. And when we get a base level of fitness and strength built up, we need you to do maybe some little box jumps and try to do some explosive movements and do some sprinting. Because, I mean, most people, unless they're an athlete, they're probably not sprinting after their 20s, maybe into their 30s. They're just, the family comes, the job. And so they're not producing those explosive movements. They may not be weight training heavy and recruiting these type 2 muscle fibers. And so, like you said, if you don't use them, you lose them. But then this has implications when you're 70, 80, 90 years old. It may be very difficult getting up and down steps, and it may be very difficult doing those activities of daily living because you have trouble recruiting or you've lost those type 2 fibers that really allow you to produce a lot of force and, live an independent life where you're not dependent on somebody.
I agree with you.
Okay, well, great. On to the next slide, right? Slide 13.
So here's just one example, could be many of them, but this is a chance where we had international level weightlifters. So the sport of Olympic weightlifting, those of you not familiar with it, it's the snatch and the clean and jerk. And both of those lifts are very rapid. You cannot do them slowly, just like you cannot do a jump slowly. If I'm in the sport of powerlifting, I try to move the weight as fast as I can, but the masses are so heavy that you usually grind through those very heavy lifts. I've
always thought powerlifting is a bit of a misnomer, but I mean, that's just what it's called.
It is. It's a mass lifting, but that doesn't sound as cool.
Right.
But
because Olympic lifting, like you said, you can't move a bar from off the ground, to over your head slowly. So that should be called Olympic lifting. Should be called powerlifting, right? Force times velocity.
Yeah, don't get into that discussion.
Right.
But this does let us look at some unique characteristics. So a number of years ago, we were funded by the USA Weightlifting, which is the national governing body for the sport here, and they're based out at Colorado Springs at the training center out there. And so we actually did a road trip up to one of the regional training centers the week after nationals. And so all the individuals we got biopsies for, it wasn't going to interfere with their training for national meet. We knew they were in peak performance, their training had peaked. And so then we also had some healthy exercise science students that served as controls. So on the left versus the right, left is the controls, right is just one of the weightlifters. You can just visually see the fibers are larger. You can see there's a lot of 2As. On the right, there are no 2 back then we called them Bs or ABs. There's like one AB hybrid. In this case, there was no 2Xs in the 1,000 fibers whatsoever, because they had trained those away. And so it's just a lot of these are things I am sure was due to their training, the muscle growth, what is an optimal size. The vastus lateralis is a perfect muscle to study in this sport because so much of what they do, their lower body strength is
knee extension
and power is critical. And so this was just one of several studies we did. We did a similar study with national and international level power lifters and saw some similar results.
So a lot more 2A fibers and larger fibers, all of them,
Yep.
in the international caliber weightlifters, and then the powerlifting study you did.
Yep.
Yeah.
So,
and that was back in 2003 you did that. There's the Fry and colleague.
Yeah, and we actually still have more data that we may go back and do some secondary analyses, because the way we preserve these slides, they're still viable. You come in our lab, and because depending on how you preserve them, sometimes after a few years they're not really viable to analyze.
Now then there's the question: are they international caliber weightlifters due to their genetics, or is this due to training? Is this a training effect?
Good question. Well, their parents didn't let us come and do muscle biopsies from them when they were in preschool. And and what's their activity history prior to becoming weightlifting? Because quite often they come out of other sports. Some of them got into weightlifting early on. Some of them gravitated to it after they finished a football career, or they were a track athlete. I mean, you get a wide mix. And although in some countries you get people started in the sport of weightlifting early on. So I mean, it really kind of
like
depends.
They do in China. Oh my gosh, they start them young.
And so and we actually have some interesting data here, and it's related to their performances. So we have a lot of data we won't get into today, but people talk about how important is the fiber type. Sometimes it's hard to see correlations between actual performance in an intact human and the data from their biopsy, but there's plenty. It kind of depends on what you were measuring and how you were measuring it, and what your analysis of the biopsy was.
Right, and it's more than just a fiber type. What fiber type you have, your ability to succeed at whatever given sport. I mean, it's multifactorial, like a lot of things. I mean, the training you do. So it's like it's not just genetics, even though those do play a role. It's the training you do, and the diet, and the sleep, and hand-eye coordination, and just your reaction time, and your experience, understanding the game, the sport. So it's
just one little
so
piece
many
of
people,
this big puzzle.
right? Yep. So many people just want to say, and they do this in some countries. They'll fiber type young kids and say, oh, well, they're mostly fast twitch, or they're mostly slow twitch. Well, if they're slow twitch, then they need to look at doing these endurance sports. Well, maybe not. I mean, depending on what their interests are, and they may want to, they may not like running or cycling for distance, even though their fiber type they may be predisposed to do better at those activities. Maybe they like the more anaerobic sports, right? So you're going to tell the child, well, no, based on your fiber type, we're pushing you this direction. I mean, it's like I said, it's one piece of the puzzle, but it's not all of the puzzle.
It helps us understand, though, or it helps us to better understand what are contributing factors to performance.
Yep. And this is one of the contributing factors. Yeah.
I think that might be it. I'm not sure.
Do we have a slide 14? Yes, we do. So I guess maybe you just want to mention anything going on in the Jayhawk Athletic Performance Lab you want to highlight right now?
Well, we are always doing a lot of fun stuff. It's always related to sport performance. As far as fiber typing, we are preparing Drake Easerhout, who's one of our doctoral students, is getting ready to continue his line of research that's on blood flow restriction. And it's going to include some biopsies, because we want to look at We're less interested in this case in the fiber type, but we will get some measures of that. We want to look at some of the other factors to maybe better explain why blood flow restriction training seems to be as effective as it is, at least in some settings. So that's coming up in the coming year. We haven't done it yet, but we're always doing fun stuff.
And aren't you writing a review on training and muscle fiber types, I thought? Or am I wrong on that? Okay.
That's it's on my desk, and there's actually two pieces of it. One is kind of doing a review of the different methods for fiber typing,
like we talked about today.
Very much based on things we're talking about and trying to help people decipher some of these different methods. And then also another one, which really deserves to be a separate one, on relationships between fiber type and performance. Particularly with intact humans, sport-related or training-related performances. Sounds easy, a little bit tricky, but there's data out there, and so trying to make sense out of a complex area.
Is there anything you want to add about the relationship between fiber type and performance, or you think we kind of covered that? It's one component or one piece in the puzzle.
I think, I think you probably summed it up best, because the human body is so amazing physiologically.
Yeah.
And this is one tissue, one organ. We have to have the nervous system that can recruit it. I have to have the ability centrally to know how to recruit it and in the right way. And there's so many facets that it would be an oversimplification to say this is the variable.
And I think that's the problem. Is I think a lot of times we as humans, we want just this simple cookie cutter one answer, that this black and white approach. And a lot of things, like you said, you get into human physiology, it's not just this or that. It's multifactorial, all these different organ systems working together in unison.
Well, a long time ago, I say a long time ago, a few years ago, former student of mine, Dr. Andy Galpin, who does a lot of good muscle work out of Cal State Fullerton, I believe he was on the Joe Rogan show and was, getting asked a lot of good training questions and so forth. And if I recall correctly, a very common response was, well, it depends.
Yeah.
That's a great question, but it's not a simple answer. It kind of depends on who you're talking about, what the training is, what the measure is. There's a lot of factors, and so that can be frustrating, but that's the way we're built.
Yeah, some of the smartest people out there are the ones that say it depends a lot, or that it's not because they don't know anything. It's just, it's hard to say conclusively until you have enough evidence that's piled up, and you don't want to give people the wrong perspective by saying, oh yeah, it's this or that. And then, years later, it's like, well, all this new research came out, and what are you talking about? You said yes. So it's like, you have to be very careful about what you say and how you say it. And some people are not.
I think you've tapped me out on everything
right.
I can say.
I think we're good. How can people contact you if they have questions about your research, teaching, the Jayhawk Athletic Performance Lab, any of those things?
Well, the easiest is certainly through my email, which is acfry@ku.edu. You can also check out the activities of our lab on social media. We have accounts on Instagram and Facebook and X, formerly Twitter, and on LinkedIn. And we try to post probably a couple times a week the various activities that are going on in the lab, and you can kind of stay on top of what we're up to.
All right, check out those social media accounts. And thanks for coming on the show again today, Dr. Fry. If you have questions for me about the KU Exercise Science program, send an email to jtaylor@ku.edu or call 913-897-8516. I hope you enjoyed today's episode, and thanks for watching.